Biochemistry. Molecular Part 1. Chromatin structure. DNA exists in the condensed chromatin form to fit into the nucleus. DNA loops twice around a histone octamer to form a nucleosome, beads on a string. H1 binds to the nucleosome and to the linker DNA, thereby stabilizing the chromatin fiber. DNA has negative charge from phosphate groups. Histones are large and have positive charge from lysine and arginine. DNA and histone synthesis occurs during S phase. Mitochondria have their own DNA, which is circular and does not bind histones. Heterochromatin condensed appears darker on electron microscope, sterically inaccessible, thus transcriptionally inactive. Heterochromatin has increased methylation and decreased acetylation. Heterochromatin is highly condensed, hidden chromatin. Bar bodies, inactive X chromosomes may be visible on the periphery of nucleus. Euchromatin. Euchromatin is less condensed, appears lighter on electron microscope. Transcriptionally active, sterically accessible. Euchromatin is truly transcribed. Euchromatin is expressed. DNA methylation reversibly changes the expression of a DNA segment without changing its sequence. Involved with aging, carcinogenesis, epigenetics, genomic imprinting, transposable element repression, and X chromosome inactivation, lionization. DNA is methylated in imprinting. Methylation within gene promoter (CpG) islands typically silences gene transcription. CpG methylation makes DNA mute. Dysregulated DNA methylation is implicated in Fragile X syndrome. Histone methylation usually causes reversible transcriptional suppression, but can also cause activation depending on location of methyl groups. Histone methylation mostly makes DNA mute. Lysine and arginine residues of histones can be methylated. Histone acetylation. Removal of histone's positive charge, which leads to relaxed DNA coiling, then increased transcription. Thyroid hormone synthesis is altered by acetylation of thyroid hormone receptor. Histone acetylation makes DNA active. Histone deacetylation. Removal of acetyl groups, which leads to tightened DNA coiling, then decreased transcription. Histone deacetylation may be responsible for altered gene expression in Huntington disease. Histone deacetylation deactivates DNA. Nucleotides. Nucleoside. Base plus deoxyribose, sugar. Nucleotide. Base plus deoxyribose plus phosphate. They are linked by 3,5-phosphodiester bond. Nucleotides have 3 components. 5 end of incoming nucleotide bears the triphosphate, energy source for the bond. Purines, Ag, has 2 rings. Pyrimidines, Cut, has 1 ring. Pure as gold, cut the pyramid. Thymine has a methyl group. Cg bond, 3 H bonds, is stronger than At bond, 2 H bonds. Increased Cg content leads to increased melting temperature of DNA. Cg bonds are like crazy glue. Deamination reactions. Cytosine to uracil, adenine to hypoxanthine, guanine to xanthine. 5-methylcytosine to thymine, uracil found in RNA, thymine in DNA. Methylation of uracil makes thymine. Amino acids necessary for purine synthesis. Cats purr until they gag. Glycine, aspartate, and glutamine. De novo pyrimidine and purine synthesis. Various immunosuppressive, anti-neoplastic, and antibiotic drugs function by interfering with nucleotide synthesis. Pyrimidine synthesis. Leflunomide. Leflunomide inhibits dihydroorotate dehydrogenase. 5-fluorouracil and its prodrug capecitabine. 5-fluorouracil and capecitabine form 5-fluorodeoxyuridylate, which inhibits thymidine synthase, which leads to decreased deoxythymidine monophosphate. Purine synthesis. 6-mercaptopurine and its prodrug azathioprine. 6-mercaptopurine and azathioprine inhibit de novo purine synthesis, which inhibit the enzyme guanine phosphoribosyltransferase transferase. Azathioprine is metabolized via purine degradation pathway, and azathioprine can lead to immunosuppression when administered with xanthine oxidase inhibitor, allopurinol, and febuxostat. Mycophenolate and ribavirin. Mycophenolate and ribavirin inhibit inosine monophosphate dehydrogenase. Purine and pyrimidine synthesis. Hydroxyurea. Hydroxyurea inhibits ribonucleotide reductase. Methotrexate, trimethoprim, and pyromethamine. Methotrexate, trimethoprim, and pyromethamine inhibit dihydrofolate reductase, which leads to decreased deoxythymidine monophosphate. In humans, methotrexate, bacteria trimethoprim, and protozoa pyromethamine. Carbamoyl phosphate synthetase 1, mitochondria, urea cycle, found in liver. Carbamoyl phosphate synthetase 2, cytosol, pyrimidine synthesis, found in most cells. Purine salvage deficiencies. Adenosine deaminase converts adenosine to inosine. Cladribine and pentostatin inhibits adenosine deaminase. Adenine phosphoribosyltransferase transferase converts adenine to adenosine monophosphate. Hypoxanthine guanine phosphoribosyltransferase transferase converts guanine to guanosine monophosphate and hypoxanthine to inosine monophosphate. Xanthine oxidase converts hypoxanthine and xanthine to uric acid. Urate oxidase, rasburicase, converts uric acid to allantone. Allopurinol and febuxostat inhibit xanthine oxidase. Adenosine deaminase deficiency. Adenosine deaminase is required for degradation of adenosine and deoxyadenosine. Decreased adenosine deaminase can lead to increased deoxyadenosine triphosphate, which leads to decreased ribonucleotide reductase activity, decreased DNA precursors in cells, and decreased lymphocytes. Adenosine deaminase deficiency is one of the major causes of autosomal recessive, severe combined immunodeficiency. Lesch-Nyhan syndrome. Defective purine salvage. Deficient or mutated hypoxanthine guanine phosphoribosyltransferase transferase, which leads to decreased guanosine monophosphate and decreased inosine monophosphate formation. Guanosine monophosphate is from guanine, inosine monophosphate is from hypoxanthine. Lesch-Nyhan syndrome leads to compensatory increase in purine synthesis, increased PRPP amidotransferase activity, which leads to excess uric acid production. Lesch-Nyhan syndrome is X-linked recessive. Findings include intellectual disability, self-mutilation, aggression, hyperuricemia, red-orange sand, sodium urate crystals in diaper, gout, dystonia and macrocytosis, HGPRT, hyperuricemia, gout, pissed off, aggression, self-mutilation, red-orange crystals in urine, tense muscles, dystonia. Treatment is allopurinol or febuxostat. Genetic code features. Unambiguous. Each codon specifies only one amino acid. Degenerate, redundant. Most amino acids are coded by multiple codons. Wobble hypothesis. First two nucleotides of codon are essential for anticodon recognition, while the third nucleotide can differ. Exceptions are methionine and tryptophan are encoded by only one codon. Homologous non-overlapping. Read from a fixed starting point as a continuous sequence of bases. Exceptions are some viruses. Universal. Genetic code is conserved throughout evolution. Exception in animal mitochondria. Some codons specify different amino acids than in non-mitochondrial DNA. DNA replication occurs in 5' to 3' direction in continuous and discontinuous Okazaki fragment fashion, semi-conservative, more complex in eukaryotes than in prokaryotes but shares analogous enzymes. Origin of replication, particular consensus sequence in genome where DNA replication begins, may be single prokaryotes or multiple eukaryotes. AT-rich sequences, for example, TATA box regions, are found in promoters and origins of replication. Replication fork, Y-shaped region along DNA template where leading and lagging strands are synthesized. Helicase, unwinds DNA template at replication fork. Helicase halves DNA, deficient in Bloom syndrome, BLM gene mutation. Single-stranded binding proteins, prevent strands from reannealing or degradation by nucleases. DNA topoisomerases, creates a single or double-stranded break in the helix to add or remove supercoils. As needed due to underwinding or overwinding of DNA, single break, topoisomerase 1, double break, topoisomerase 2. In eukaryotes, irinotecan and topotecan inhibit top1, etoposide and tenneposide inhibit top2. In prokaryotes, fluoroquinolones inhibit top2 and top4. Top2 also called DNA gyrase. Primase makes RNA primer for DNA polymerase III to initiate replication. DNA polymerase III prokaryotes only elongates leading strand by adding deoxynucleotides to the 3' end. Elongates lagging strand until it reaches primer of preceding fragment. DNA polymerase III has 5' to 3' synthesis and proofreads with 3' to 5' exonuclease. Drugs blocking DNA replication often have a modified 3' end hydroxy group, thereby preventing addition of the next nucleotide. Chain termination. DNA polymerase I, prokaryotes only, degrades RNA primer, replaces it with DNA, same functions as DNA polymerase III, also excises RNA primer with 5' to 3' exonuclei. DNA ligase catalyzes the formation of a phosphodiester bond within a strand of double-stranded DNA, joins Okazaki fragments. Ligase links DNA. Telomerase, eukaryotes only, a reverse transcriptase, RNA-dependent DNA polymerase that adds DNA to 3' ends of chromosomes to avoid loss of genetic material with every duplication. Upregulated in progenitor cells and also often in cancer, downregulated in aging and progeria. Telomerase tags for greatness and glory. DNA repair, double-strand repair, non-homologous end joining, brings together two ends of DNA fragments to repair double-stranded breaks. Homology not required. Part of the DNA may be lost or translocated. Homologous recombination requires two homologous DNA duplexes. A strand from damaged dsDNA is repaired using a complementary strand from intact homologous dsDNA as a template. Defective in breast ovarian cancers with BRCA1 or BRCA2 mutations, and in certain types of Fanconi anemia. Restores duplexes accurately without loss of nucleotides. Single-strand repair. Nucleotide excision repair. Specific endonuclease remove the oligonucleotides containing damaged bases. DNA polymerase and ligase fill and reseal the gap, respectively. Repairs bulky helix-distorting lesions, for example, pyrimidine dimers. Occurs in G1 phase of cell cycle. Defective in xeroderma pigmentosum. Inability to repair DNA pyrimidine dimers caused by UV exposure. Presence with dry skin, photosensitivity, and skin cancer. Base excision repair. Base-specific glycosylase removes altered base and creates AP site, a pyrinic pyrimidinic. AP endonuclease cleaves 5' end, removing one or more nucleotides. AP lyase cleaves 3' end. DNA polymerase beta fills the gap. DNA ligase seals it. Occurs throughout cell cycle. Important in repair of spontaneous toxic deamination. Gel please. Mismatched repair. Mismatched nucleotides in newly synthesized strand are removed and gap is filled and resealed. Occurs predominantly in S phase of cell cycle. Defective in Lynch syndrome. Hereditary non-polyposis colorectal cancer. Mutations in DNA degree of change. Silent, less than missense, less than nonsense, less than frameshift. Single nucleotide substitutions are repaired by DNA polymerase and DNA ligase. Types of single nucleotide point mutations. Transition purine to purine, or pyrimidine to pyrimidine. Transversion purine to pyrimidine, or pyrimidine to purine. Single nucleotide substitutions. Silent mutation. Codes for same synonymous amino acid, often involves third position of codon, tRNA wobble. Missense mutation. Results in changed amino acid, called conservative if new amino acid has similar chemical structure. Examples: sickle cell disease, substitution of glutamic acid with valine. Nonsense mutation. Results in early stop codon, UGA, UAA, UAG. Usually generates non-functional protein. Stop the nonsense. Other mutations. Frameshift mutation. Deletion or insertion of any number of nucleotides not divisible by 3, or if divisible by 3 split across adjacent codons, which leads to misreading of all nucleotides downstream. Protein may be shorter or longer and its function may be disrupted or altered. May occur due to slippage of DNA polymerase during replication at repetitive nucleotide regions. Examples: Duchenne muscular dystrophy, Tay-Sachs disease, cystic fibrosis. Splice site mutation. Retained intron in mRNA, which leads to protein with impaired or altered function. Examples: rare causes of cancers, dementia, epilepsy, some types of beta thalassemia, Gaucher disease, Marfan syndrome. Slipped strand mispairing. Occurs when DNA polymerase slips and either inserts or removes one or more additional nucleotides by mistake in an area of repetitive nucleotides. Anticipation occurs secondary to insertion of increased repeats across generations. Example: CAG repeat expansion in Huntington disease. Lack operon. Classic example of a genetic response to an environmental change. Glucose is the preferred metabolic substrate in E. coli, but when glucose is absent and lactose is available, the lack operon is activated to switch to lactose metabolism. Mechanism of shift. Low glucose state. Increased adenylate cyclase activity, increased generation of CAMP from ATP, which leads to the activation of catabolite activator protein, which leads to increased transcription. High lactose state. Unbinds repressor protein from repressor operator site, which leads to increased transcription. Low glucose lactose available. LAC genes are strongly expressed. High glucose lactose unavailable. LAC genes are not expressed. Low glucose lactose unavailable. LAC genes are not expressed. High glucose lactose available. Very low basal expression of LAC genes.